Hi and hello everyone. This is our lab report for practical 2, which is about factors that affect the distribution and abundance of organisms. So this is our group members. For the introduction, ecology is sometimes defined as a measure of the distribution and abundance of organisms. With slow-moving or non-moving organisms, the quadrant survey technique can be effective. A traditional method for studying ecology, particularly biodiversity, is quadrant sampling. It is a crucial technique for directly counting organisms in a specific percentage of the ecosystem. Population distributions, density, frequency, and abundance are all estimated using it. In plant studies, the quadrant method has been applied extensively.
The objectives of these experiments are to introduce the simple concept of quadrat, to enhance the power of observation, to record the difference in plant distribution within the quadrat, and the last one is to infer from natural observation the possible factors that affect distribution and abundance of organisms. There are 5 materials that are needed for this experiment. The first one is raffia strings, the second one is stick, the third one is measurement tape, the fourth one is scissors, and the last one is notebook.
For the first one, different types of plant species at UiTM campus JENGKA forest will be the area under study. 10 quadrats of 5 meter multiplied 5 meter were made. Multiple quadrat sample will be performed throughout the habitat at several random locations. All the tree stands that are found inside the quadrat were observed. The data will be used to estimate the population size and population density within the entire habitat.
These are the results that were taken from practical 2.
From the data collected, it shows that species Y has the most number of individuals, which are 12 individuals, following with species D and species AB, both are 11 individuals. Furthermore, quadrant 4 has more abundance and distribution of organisms than the other quadrants. To make it understandable, the variation of species across quadrants are in uneven distribution. This is because some species are mostly absent in some quadrant or low in abundance. The different number of plant species in the quadrant occur due to the abiotic factors and some biotic factors. Abiotic factors are the non-living factors which affect the ability of living organisms to survive in an environment. This can include both of physical and chemical properties. Biotic factors are factors produced by living organisms that affect the ability of other living organisms to survive in an environment. The first abiotic factor is the soil composition. When there are variations in soil nutrients, pH, and texture within the quadrats, cause differences in plant species distribution. Areas with richer soil in terms of organic matter and nutrients might support a higher diversity of plant species compared to areas with poorer soil quality. Light availability also one of the abiotic factors that affect the plant distribution and abundance. As light is a crucial factor for photosynthesis, the differences in light intensity and duration across the quadrats influence plant species diversity. Quadrats that receive more sunlight might have a higher number of species that thrive in well-lit conditions, while shaded areas might support shade-tolerant species. Variation in canopy cover can create these differences. Moreover, moisture levels is the abiotic factor when variation in soil moisture content across quadrates. This is because soil moisture influences the types of plant species that can thrive. Some plants require consistently moisture conditions, while others are adapted to drier environments. Differences in topography, drainage, and water retention can cause variation in moisture levels. Seed dispersal and germination is the biotic factor. This occurs when variation in seed dispersal mechanisms and germination success affect plant species distribution in the quadrates. The presence of certain species may depend on how effectively seeds are dispersed to and germinate in specific areas. Factors such as wind patterns, animal movement, and the presence of the seed predators or dispersers, such as insects, can influence seed dispersal and establishment. Lastly, the biotic factor is the competition and succession. Differences in interspecies competition and ecological succession stages influence the plant species present in the quadrates. Some quadrates might be at different stages of ecological succession, leading to variation in species composition. Additionally, competitive interactions between plant species for resources, in particular light, water, and nutrients, result in dominant species outcompeting others, affecting diversity. For conclusion, overall, quadrant sampling showed basic idea of a quadrant by doing a deep observation. This allows for detailed study of a manageable portion of the habitat, reducing the amount of time and resources needed. The distribution and abundance of organisms are determined by a complex interplay of biotic and abiotic factors. By understanding these factors helps in conserving and preserving the biodiversity.
For the post-lab question, first, define the term quadrat in practice. Quadrat is defined usually square, plot used for sampling in field study to assess the distribution and abundance of organisms within a specific area. Quadrats are fundamental tools in studying and monitoring plant populations, particularly in ecosystems such as forests, grassland, and intertidal zones. Second, what are two basic requirements of all the quadrat techniques? First, randomly select locations within a meadow to place the quadrats, ensuring that different parts of the meadow are sampled proportionally. Second, consistently use the 1 meter square quadrats at each location to count and record the plant species and their abundance. Third question, what would be the advantages and disadvantages of using a larger or smaller quadrat to survey the sedentary organism? Larger quadrats are beneficial for capturing a broad, representative sample of a community, reducing variability and minimizing age effect, but they require more time and effort to survey. Smaller quadrats offer efficiency and detailed observations, but can result in higher variability and may not be as representative of the overall community. The last one, problems with spatial scale often make it difficult to directly estimate average densities in habitats such as large forest area or in aquatic habitats where one cannot directly see the animals or plants of interest. Explain the issues concern ecologists in conducting the sampling method. Ecologists face significant challenges in estimating average densities in large forest area or aquatic habitats due to issues related to spatial scale and visibility. In vast forested area,
The heterogeneity of the environment complicates sampling efforts. The diverse microhabitats, variations in vegetation density, and terrain can lead to highly variable species distributions, making it difficult to ensure that samples are representative. Random sampling techniques might not capture the full diversity or density of species present, resulting in below the surface or in murky water, obscuring direct observation. This necessitates the use of indirect methods like remote sensing, sonar, or trapping, which can introduce their own biases and limitations. Additionally, the dynamic nature of aquatic environment with factors like water, currents, and seasonal change further complicates the accurate estimation of densities. This issue requires ecologists to employ a combination of innovative sampling technique, rigorous statistical method, and adaptive strategy to obtain reliable density estimate in such challenging habitat.